So in the long term within PERSIST-SEQ, we will deepen our understanding of evolving resistance in different types of solid cancer across different targeted agents. Based on this knowledge, the combination of different treatments can be optimized also in the clinic to reduce the likelihood of recurrence early in the treatment history of a patient. My name is Julia Schüler, and I'm the Therapeutic Area Lead Oncology in Charles River. In the consortium, I'm a PI and a work package leader for the PERSIST-SEQ. So what really thrills me to be part of this consortium is that single-cell sequencing is one of the emerging technologies that have the potential to help us better understand cancer as a disease in general, but also shed light into specific questions such as resistance mechanism, for example. However, with many new technologies, there's the need for a validation and the development of robust protocols. And the work of this consortium will enable us to make the most out of this new technology, single-cell sequencing, and also of the data sets, and to get a deeper understanding of the underlying biology of resistance mechanisms, one of the most challenging topics in cancer research. Very basically, a PDX, a patient-derived xenograft, is a human tumor tissue from a biopsy or a surgery that is then implanted into an immune-compromised mouse. In case the tumor tissue is growing, it is then transplanted into another mouse, and by this approach, you can generate almost endless amounts of tumor tissue that represents a specific patient. This tissue can then be used for study tumor biology or for drug development. The difference to an organoid is that it's obviously used in an animal and not in a cell culture, and that you have all cell types that are part of a tissue involved, although the only human cell in that PDX is the tumor cell, and all other cells will come from the mouse. Each PDX model is characterized by several different technologies. We are starting from basic histology, but also RNA sequencing or whole exome sequencing data are generated and then compared to the biopsy of the patient. So this is how we compare or make sure that the model we are creating represents the patient. Across a panel of models that represent a specific tumor type, for example, non-small cell lung cancer, these data are all combined and then compared to data from patient cohorts of the same tumor type. We and others have shown that these PDX panels represent very closely the molecular as well as the phenotypic landscape of the respective disease. So the work package 4 also ensures the alignment on quality controls for the samples that will be analyzed during the project. And furthermore, a consensus for sample labeling was achieved and a constant flow of incoming samples was established. These are rather operational details that ensure the comparability of the different samples across the consortium now and also in the future. So specifically when dealing with big data, such as single-cell sequencing data, this is crucial to have the data annotated in a very clear and unambiguous way. The milestones we have achieved in Work Package 4 were starting from contributing expertise in protocol and workflow development. We now are generating samples, in our case from non-small cell lung cancer PDX, to contribute to the consortium database. And within Work Package 4, we have reached our milestones in time and can provide a workflow of single-cell sequencing acquisition that then enables the comparison of all generated data across the different cell sources, as well as different time points during the consortium funding period. So challenges of developing and maintaining PDX are, for example, the fact that PDX models grow relatively slow compared to other models used in cancer research, and obviously need the use of lab animals. So timing can be very challenging as you want to have the material at hand in time, but also do not want to use more animal than absolutely necessary. A huge improvement for us was the possibility to use frozen sample instead of viable material for the single-cell sequencing, which allowed us to run the in vivo experiments independent of the availabilities of the groups that are performing the sequencing. So the comparison of the different models across drug discovery for oncology is that for the in vivo experiments in cancer research, the PDX are the gold standard based on some of the characteristics I mentioned earlier, but basically because of their ability to represent a patient cohort of the respective disease. In comparison, organoids and cell lines are important platforms used in vitro and have the advantage of being fully human. PDX tissue, on the other hand, always incorporates mouse cells, such as fibroblasts or immune cells, and all the different model systems used in the consortium cover different aspects of the disease. So the choice of your model depends on the scientific question you want to answer. Most of the time, you will use a combination of different models. So we are collaborating with many partners across the consortium. With VHAO, for example, we share the work package lead. With AstraZeneca and single-cell discovery, we are piloting the analysis of the xenograft samples. And through the different work package meetings, we are in close contact with a number of academic and industry partners. The feasibility to analyze the PDX as well as the organoids and cell line material is still relatively new technology. The acceptance of it increased in our group. So beyond PERSIST-SEQ, we will include single-cell sequencing also in other projects around tumor plasticity under different culture conditions. The publication that really excites us was the manuscript describing the gene expression flex protocol as valid for xenograft samples, which is seen as a major step for our studies using PDX material. So without the input of samples and expertise from the different labs, this publication would not have been possible and showcases also the strength of the consortium, such as PERSIST-SEQ. So GDPR, animal welfare, and patient-informed consent are essential components of ethical and legally compliant scientific research. GDPR protects individual personal data, ensuring transparency and privacy in how much information is collected, stored, and used. For animal welfare standards, the safeguard is the humane treatment of the animal in research, not only addressing ethical concerns, but also improving the reliability of the scientific results, which is very important. And last but not least, the patient-informed consent ensures that the human participants are fully aware of the study's purpose, risk, and benefits, and that their participation is voluntarily. Together, all these principles uphold the integrity of our scientific research, promoting public trust as we are using public money, and ensure adherence to legal and ethical standards. So in the remaining time of the project, once we have analyzed the data that are coming from our lung cancer PDX models and hopefully have identified a few key drivers of persistence in this setting, we will verify these analyses by modulating the identified escape pathways, either pharmacologically or by genetic modification, to mechanistically showcase the relevance of our findings. And in addition, the comparison of our dataset with other lung cancer models from organoids or cell lines or even patient samples will help to get a better understanding of the biology behind those data.